Hi everyone. Let me start by thanking the organizers for giving me a chance to be here and also for putting together this program, which has been a great experience so far. My name is Mathieu. I work as an application scientist with IBSBC, and what I won't do is I won't talk about any hardware introductions or something like that. Instead, I'll do is I want to talk about a collaboration that we started about a year ago with the ETH and Zurich. So most of the results or of the developments you're going to see are actually originating there. It's going to be a bit of a sneak peek or a work in progress, if you wish. What is this all about? It's about a platform called NMArtist, which is a fully automated cloud platform solving protein structures. And even though this is the first slide, this is where I expect some of you start rolling their eyes, because let's face it, solving static structures of individual singular proteins may not be the best use for NMR or pretty much any other experimental technique these days. So why would you still do this? And let me start by some of the challenges. And so basically the idea behind determining 3D structures by NMR is taking information such as the primary amino acid sequence and a series of spectra, usually higher dimensionality, 3D, 4D spectra, and turning them into a structure. And now the question is how to get there, and more importantly, how to get there fast. So in some cases, you just want to get this done as soon as possible. Maybe because this is you working on a competitive project, maybe this is because the protein is part of a disease that's currently screwing up the world around you, or maybe just because you're impatient. So what do we need for that? First of all, we need a way to produce a sample. Typically, we deal with isotopically labeled samples. Most of the time, carbon-13, nitrogen-15 labels, so stable isotopes. In some cases, deuterated, and this takes a bit of experience in terms of sample production. Also, we work with slightly larger sample amounts as compared to some of the other techniques. Once you end up with a sample, you put this in a small glass tube and you take it to the spectrometer. So that would be step one, and that is something that has been covered for a long time now. Now the next what you need is, and you need to know that so far, determining a structure by NMR is mostly a manual process, and it involves a person that is willing to do this. Ideally, this is it's a dedicated person, and it's also a person that doesn't easily get bored. So for the lack of a better example, I'll just put younger myself down there. And also that person is going to need some training. And if you really want to push this, if you really want to get this done fast, well, the calculations involved, yes, you could do this on a regular workstation. However, if you go through multiple iterations and you're in a rush, then you're going to require some additional computational power in order to get this done faster. And as I said, if you really want to push it, you might need some help in one form or another. And also, the poor guy down there better have a couple of years of experience. Now, why is that? Why is this such a labor and time-consuming task? And if you just look at a very general workflow of how to get from the wet lab to a result, which in this case, it's a 3D structure, but this holds true for a lot of other applications. Whether this is the characterizing conformational chemical exchange, the workflow is pretty much the same. And so what we have been covering for a long time is guidance or automation when it comes to experimental setup, data acquisition, data processing and post-processing. What still remains, in my opinion, a major bottleneck is resonance assignment. We usually like to make this claim that everything we do with NMR is at atomic resolution. And while that's true, in order to link the information you gain to a particular atom, you need to execute this process called resonance assignment. This usually involves a person sitting in front of a screen and looking at multiple 3- and 4-dimensional spectra, cross-referencing them, going back and forth in between, which is a very tedious task, and it's also not the most fascinating one. What we can do about this? So what is NMArtist? I mentioned before, NMArtist it's a cloud-based platform that automates peak picking, resonance assignment, and the analysis of protein spectra. For example, if you wanted to perform structure determination. It's a combination of traditional combinatorial approaches as well as more modern or more recently developed approaches utilizing AI. And the main idea is that it really saves time, it really reduces the effort. It takes sort of the boring tasks off your hand, and it should also enable a person that doesn't have years of experience to, for example, determine the structure or work with NMR on a protein in another way. And so the underlying platform and goes by the name of Artina, so this is the name by which this has been published, and it consists of sort of 3 steps that have to be or can be executed in consecutive order. The first one is the peak picking, and this is where the group at the ETH Zurich really did an amazing job. So they curated about 3,000 higher dimensionality spectra in order to use this as a training data set. The next step is the shift assignment, and that is sort of a 2-step approach. One is the backbone assignment, which is a traditional combinatorial approach, if you want, and then they trained another network in order to perform sidechain chemical shift predictions in order to do sidechain assignments. And then, if you want to, you can follow up by doing a structure determination. Now, usually what I would do is, at this stage, I would show you an example of ubiquitin, which is our favorite pet molecule that nobody cares about, and show you how well this works. And I've done this in the past. And then I was approached by one of our customers who was, let's say, a little bit suspicious, and he basically told me nobody cares about ubiquitin. I mean, how about I prepare a sample which is more realistic? I bring it, we run the measurements together, and we see what it can do. So he showed up with a sample. It's a cancer-related protein, just shy of 20 kilodaltons. Was a 400 micromolar solution, uniformly carbon and nitrogen labeled, non-deuterated, and we did all the measurements at an 800 megahertz spectrometer with a cryoprobe. So yes, high field, but nothing crazy. And the majority of the measurements have been set up, processed, and referenced in full automation. And what you can see here on the left-hand side is the results of the automated assignment, peak picking and assignment. So here on the top, you can see the performance of the individual spectra in terms of peak picking when comparing it to the benchmark spectra. And in the bottom, you can see the sequence plot, and you can see using a threshold. We have dark blue for high confidence, and we have light blue for lower confidence. And in this case, we were able to work with all the spectra and end up with roughly 91% predicted to be accurate sidechain 91% predicted to be accurate assignments. Now we followed up by performing the automated structure calculation. But first, let me show you. This is a list of the experiments we used. And without going into too much of the details, we do have a bunch of experiments that are only used for the assignment, and then we have two of the experiments that provide you with actual distance restraints, which are then used for the structure calculation. And what you can see here on the right-hand side is the overlay of the 20 lowest NSC structure from the NMR test run, and on the left-hand side, you can see the crystal structure of not the exact same construct. So the amino acid sequence was not 100% identical. Also, you can see that, of course, some of the loops are missing, but we were pretty happy with that, and this is exactly what they expected to come out of something like this. Now, what we're also doing, we were having a look into sort of what combinations of experiments work best. And I just want to show you just one slide about this very briefly. And this is not my desperate attempt to sneak some NMR methodology into there, but this is the kind of details we're looking at. So this is one of the, if you wanted to perform structure calculation, an experiment called the 3D NILSI is one of the experiments that gives you distance restraints. So that is crucial for determining a structure. And as you may know, there is hundreds and thousands of NMR experiments. They come with different information content. And a lot of these would also come in sort of different flavors if you want. So some better suited for small molecules, some better suited for larger molecules, some working better or worse depending on the labeling pattern. And what we're trying to do right now is really to come up with a reasonable suggestion of combinations of experiments that do work best in that particular context. So for example, looking at the spectrum, and if you haven't seen this before, trust me, this is about the most simple sequence that you could run that would provide you with this information. If you have dealt with the spectra before, I hope you can appreciate the spectrum quality. So this would be a two-dimensional projection showing all the proton-carbon correlations. This would be an individual slice, and all these cross signals that you see down here. These are the ones that actually provide you with the distance restraints. However, there is different ways how you could run these experiments. However, when trying to run this as easy as possible, as straightforward as possible, you're always making some sort of compromise. Usually it's about sensitivity, spectrum quality, robustness to, for example, miscalibration, and you're usually doing some sort of compromise for all of these. And in that case, we would be willing to sacrifice a few percent of sensitivity in favor of choosing a more robust experiment. Also, if you want to analyze these automatically, you might want to go with a spectrum that has the potential to produce rather the cleanest spectrum over the one that gives you the most sensitivity. So this is the kind of details that we're currently looking at, and what we want to do in the end is we really want to provide a reasonable suggestion of experiments that do work best for a given combination of molecular size, labeling scheme, and I think we're getting there. Right. As I said, I think the true potential of this is not necessarily solving structures. While this is nice, and there's probably always going to be the one odd example where no other technique is going to get you there, then you'll revert to NMR. However, I think there's better uses for this, and as you have seen in this three-step approach, one of the byproducts is the resonance assignments. So, and forgive me for showing a slide like this. Why should we look at dynamics by NMR? Well, there's plenty of reasons. Over the years, people have developed all these beautiful experiments that can cover an entire range of time spans. And it's not just because we can, but there's also actual processes happening on those timescales. But I think there's one quote from one of yesterday's lessons that probably summarizes this best. If it doesn't move, it's probably not that interesting. And forgive me for moving back to ubiquitin. So this is done on our PET molecule. So what I did is I used what I thought would be a good minimal set of experiments. So basically, the combination of these two three-dimensional experiments would provide you with backbone assignments. Experimental time, combining a couple of techniques for speeding up the experiments, was just under 2 hours. Calculation time for the AI-based peak picking and the backbone assignment, 35 minutes. And we ended up with 98.5 correct backbone assignments in that case. And then we took these, we ran a bunch of relaxation experiments, again in full automation, and we took this to our in-house software that can do analysis of certain relaxation experiments, and this is the result of it. So what you can see in the back is a software called Dynamic Center. It analyzed 3 individual experiments and then combined the information in order to calculate the various arbotype order parameters. So at a first approximation, this is just a measure of flexibility. And then we used a crystal structure just to sort of overlay the color coding according to the dynamics. So again, this is our PEP molecule, which is kind of boring and rigid molecule. You can see there's really not much going on. You can see some increased flexibility in the loop regions, and you can see increased flexibility in the tail. But despite that, there's not much going on. The only point I wanted to make here is that all of this has been done in full automation. So all the experimental setup has been done by just picking, like, I want to do relaxation experiments. Processing, referencing has been done in automation. We use an NMR platform to derive the backbone assignments. And then also with basically a single push of a button, we could hand over all the acquired data to the Dynamic Center and get a result like this. And I think this is really what I consider a step forward, because ideally this would enable a lot of people to not just analyze these data. There's other ways how you can analyze these data. There's a lot of free software out there, which are mostly command line based tools that require some knowledge, also manual data in a conversion. You could end up at similar results like this even before. But I think nowadays getting to something like this is easier than ever. And this doesn't have to be just flexibility. This could be localizing chemical or transformational exchange. This could be localizing interaction patches on the surface of protein. So there's many more applications. And with that, I want to thank a couple of people, Piotr Pilkowski, and who is part of the group of Roland Rieck at the ETH in Zurich, Peter Günther, Mark Vitorino, who approached us with the cancer-related protein sample, and of course, you for your attention.
Thank you.